Hi, my name is Taylor, and this is my biomolecule. I have dihydrofolate reductase, also known as THFR, and this is an example of the molecule through mole view, just of its different secondary structures here. So DHFR is a very important enzyme. The reason it's so important is because it's essential for DNA replication, and without it, DNA replication cannot take place. And I'll elaborate on that a little later, but it's what happens or what it does is pretty simple. It basically just reduces the hydrofolate right here to tetrahydrofolate. And as you can see, the only change is the reduction of this bond right here and this bond. And so this is your primary ligand. Then NADPH or sometimes NADH can act as a cofactor for this. And it's really just because it's a proton source, and it will provide one of these hydrogens needed. The other hydrogen will be seen a little later how it's provided. So this is more on its basic structure. So the hydrofolate reductase is a dimer, so it has two of these identical monomers, and the whole structure has 186 amino acid residues, different ones through it. And so how it folds is due to just common interactions like hydrogen bonding, hydrophobic interactions. But each monomer consists of 8 beta strands and then 4 alpha helices, and you can see them. Here's your beta strands, your alpha helices, and they each have different regions that are important. So this is just one monomer. These are your subdomains that are within each. And so here's some important loops, your GH loop, your FG loop, and these are prosthetic rings that are within the loop or within the enzyme. And so these will actually serve as that source for that second proton. And so the methionine 20 loop is important because this holds that in place so that the reaction can happen more efficiently. So this is just an overview of just the ligands. Here's the structure of NADPH, and here's dihydrofolate. So again, these are the two primary ligands. This is your ligand, and this is your main cofactor just for this reaction to take place. So the ligands, again, for the protein itself, most of its interactions within its domain are hydrogen bonding and hydrophobic interactions. But for the ligands, they bind primarily via hydrogen bonding. And so those exact hydrogen bonds will be shown later and just how it compares to inhibitors. But the hydrofolate has 2 places where it can hydrogen bond, and it will hydrogen bond to this aspartic 27 residue, which is in this area. And so this will fold in to touch the prosthetic group due to via conformation change, and so that will conformation change, and that will uh, make that proton bind to it more efficiently. And then once it binds all the way in, it'll form steric hindrance that are in it, and it'll provide necessary energy. So here's a chart just of those different conformations. So it's 3 primary conformations for DHFR, and this is while reducing dihydrofolate. So it starts in an open conformation, just where this methionine 20 loop is kind of spread out from this site right here. And this just allows it to be super easy for dihydrofolate to bond. And as soon as dihydrofolate hits the active site, this methionine group will close, which will bring the dihydrofolate in contact with this prosthetic group, which there provides the second proton for the reaction for it to fully form tetrahydrofolate, as you can see here. And it's still attached in the occluded form, in the occluded conformation to the protein. And so what will happen is the methionine 20 loop will conform further in just to create steric hindrance in this region, and that will produce energy kind of to sling it out and to shoot that tetrahydrofolate out. So here's just a basic flow chart of relates to DNA. And so first, here's our enzyme, and so it comes in contact with dihydrofolate, it binds, it undergoes that whole process, shoots out tetrahydrofolate. So this is a important ligand for this synthase, thymidine synthase, and so that will form DTMP, which is an important part in DNA replication and forming DNA. So DHFR is so important because it's the only place in your body that produces tetrahydrofolate, and this means that DHFR is essential for DNA replication. And as you can see here, it's pretty far upstream from DNA, but if you eliminate this tetrahydrofolate from ever happening, then DNA can't be formed. And it's pretty important because it's in many tissues in the body. And so, according to this chart from the National Library of Medicine, this is basically an RNA sample of where DHFR can be found in cells. And the top three primary places where it was is fat, lymph nodes, and bone marrow. But it's pretty evenly distributed other than that, other than maybe the pancreas and the heart. And it's very common throughout the body. So common inhibitors, well, if DHFR prevents the synthesis of DNA, then why would we want to inhibit it? Well, it can be used in medicine. It's actually inhibiting DHFR is important for chemotherapy because it can help prevent DNA replication for rapidly produce cancerous cells. So the primary inhibitor that is used for this is methotrexate. So it acts as a competitive inhibitor because it will bond to the same active site as dihydrofolate, and it actually binds at a rate at an efficiency 1,000 times greater than dihydrofolate, and that's due to it has similar structure, but it's due to it has 3 areas where it can form hydrogen bonds compared to 2, and so that will just nearly always be favored over this. So once it binds, it will prevent dihydrofolate from binding, which will prevent tetrahydrofolate from forming. So other organisms also have DHFR, and it functions very similarly in producing tetrahydrofolate for DNA replication, but it is a little different, and it can be seen through this chart. So what this chart says, this is basically concentration of inhibitors needed for 50% inhibition of tetrahydrofolate from forming. And so here's your different inhibitors. And so this one right here is methotrexate. And so if you look throughout most of these species, it requires the least amount to reduce it to 50%. Besides guinea pigs, this BW58265 actually needs the least. But again, it's similar, but these different species do react differently to different inhibitors, or its DHFR reacts differently to different inhibitors. So that suggests that its structure is slightly different, and how it responds to these are different as well. So here are my references. Here are my questions. Just what string of effects would happen if DHFR had only the open conformation? And how would inhibition of DHFR be affected if there were 2 binding sites for different ligands? And how would this affect the approach to chemotherapy?